What is hemoglobin? Hemoglobin is a large, complex protein. It is a water-soluble globular protein. Hemoglobin's main function is to transport oxygen and carbon dioxide around the body and to the tissues which require them. Hemoglobin is made up of 4 myoglobin tertiary structures, along with a heme group attached to each myoglobin. Hemoglobin contains 2 alpha chains, which are made up of 141 amino acids, and 2 beta chains, which are made up of 146 amino acids. Each myoglobin contains 8 alpha helixes. These are the result of each polypeptide chain reacting with itself to form the most stable structure it can. The 2 alpha chains are identical, while the 2 beta chains are identical with each other. The tertiary structure of hemoglobin is a combination of the secondary structures, the alpha helixes. The heme group is extremely important in the making of the tertiary structure. The histidine residue within the heme group aids in the specific 3D structure required for hemoglobin to complete its function. Hemoglobin is organized into a tight spherical shape. The heme group of a hemoglobin is a non-protein group. This is the active site of hemoglobin. The heme is made up of a flat organic ring which is around an iron atom. The organic part of the heme group is known as a porphyrin ring, which is based off of the substance porphyrin. This ring contains a lot of conjuncted double bonds. The iron and the chain which makes up the protein modify the wavelength of absorption, and this produces the red color which is seen in red blood cells. The heme group is located in the crevice of each myoglobin molecule. It is surrounded by non-polar residues and 2 polar histidines. The iron in the heme group binds with 4 nitrogens with the porphyrin ring. This leaves 2 free sites which the iron atom can bond with. One of these sites bonds with a histidine, meaning that the last free bonding site can be used to bind the iron with an oxygen or carbon dioxide atom. A second histidine is used to modify the shape of the crevice, which allows only small molecules to enter the heme group and react with the free iron bonding site. This second histidine is also used to aid in the reverse binding of oxygen, so the oxygen can be released into the tissues. This occurs when an oxygenated hemoglobin comes near a tissue, which requires oxygen. The carbon dioxide atom in the tissue displaces the weakly bonded oxygen atom and then replaces it. When a hemoglobin is bonded with a CO2, it is then called a carboninnohemoglobin. It is due to the shape of the crevice which allows the gases to bind. The many double bonds allow only small atoms in, and the particular placing of the histidine residues allow for the binding and reverse binding of gases. Hemoglobin has polar molecules on the surface and non-polar molecules on the inside. The polar molecules are hydrophilic, which allows it to be soluble in water, so it can be transported around the body via red blood cells within the blood. Also, having hydrophilic molecules on the surface of the hemoglobin protects the hydrophilic molecules which are on the inside. This is extremely important as the heme group is connected to the myoglobin subunits through hydrophobic interactions and covalent bonds. If the hydrophilic molecules were not on the surface of the water within the blood, it would prevent these bonds from occurring. Myoglobin is the subunit within hemoglobin, but myoglobin can exist on its own. It is normally found within muscle and other similar tissues. However, myoglobin is limited as it can only bond with one oxygen atom at a time through its single heme group, while hemoglobin is able to bond with 4 atoms as it has 4 heme sites. This means that it is more suited to complete the biological function of transporting oxygen around the body, as it is able to provide tissue cells with a lot more oxygen a lot more efficiently.